Hey everyone, welcome to RadioBio Season 2. We've got several great podcasts scheduled for release this season. Stay tuned.
Today on RadioBio, evolution. No small topic. Biologists can use a diverse array of systems to try to test evolutionary concepts. Some systems, like bacteria, are useful for looking at how evolution happens in real time because they have such short generation times. But others, like animals, are much more difficult, but can allow us to ask really interesting questions like how behavior influences evolutionary processes. Today's guest, Dr. Kathleen Ferris, asks questions about how organisms respond to stress in an evolutionary sense using two very different systems. I'm Jeff Lauder.
I'm Kinsey Brock.
This episode of RadioBio of Mice and Mimulus will tell the story of organismal response to stress in 4 parts.
Don't know much biology.
Part 1, A Girl and Her Flowers.
I'm Kathleen Ferris, and I'm a postdoctoral fellow at UC Davis in the Center for Population Biology. I actually became interested in biology and specifically plant biology. Well, plants, let's be honest. I was 3. So the biology part wasn't, but I became interested very early in plants because I thought they were beautiful and amazing. So when I was about 3, I went and picked absolutely all of the hosta lilies in my mother's garden, and I got a lot of trouble. She was very angry. But at the end of my punishment, she came and told me that I should be a botanist when I grew up because then I could pick any flower that I wanted and nobody would put me in timeout. And I think that stuck with me because from then on, when people asked me what I wanted to be, I was like, I want to be a botanist. And they're like, you're 5. You don't know what that means. And I was like, yes, I do. Study plants. But I became interested in ecology and evolution and genetics specifically in college because I worked in a plant pathogen coevolution lab, and that was my first research experience at University of Chicago. And I just wanted to work on plants. I didn't really think about the questions that would drive research or anything. I just loved plants. So as a very young college student, I wrote this very famous professor an email that would be really embarrassing now, where I was like, hi, I'm Katie. I love plants. And I really would like to work in your lab.
Do you still have that email?
Oh, I'm sure I do. Maybe I don't, though, because I might not have had Gmail then. But it was very just plants. And that's not really what she did. She was an evolutionary biologist, and she just happened to use Arabidopsis as this model system to study disease and plant, coevolution.
So if you'll remember back to Season 1, we talked with Dr. Johanna Schmidt from UC Davis about her research looking at drought genetics in Arabidopsis, this small flowering plant belonging to the mustard family. This plant's considered a model organism because its entire genome is sequenced. It was actually the first plant ever completely sequenced. Arabidopsis also has a rapid life cycle, and it's relatively easy to grow in the lab, so it's perfect for studying evolution. Arabidopsis also only has 5 chromosomes in a genome that's about 125 megabases. Compare that to humans, we have 23 chromosomes and our genome is about 3,200 megabases. This makes Arabidopsis a simpler system to work with to try and understand how genes interact with the environment or code for particular traits or phenotypes.
But she hired me anyways, which I'm very grateful to her. Thank you, Joy. And then I ended up spending the rest of, 2.5 years, the rest of college, working in her lab and developed an independent project with her. And just got really excited in understanding how evolution works at the genetic level. And then she helped me figure out where to go for grad school to continue in a similar way.
So how did she move from kind of a general interest in plants to the current research that she's doing now?
I thought that genetics was a tool I wanted to use into the future to understand how things evolve in natural selection and also how things work, because that's one reason I really like biology, is because I want to know how things work and I want to be able to tell a story about that, because I also really like stories.
We do too. So we're going to break Dr. Ferris's research down into 3 parts: the evolution of Mimulus, mice, and what it all means. Part 2, Mimulus.
Dr. Ferris's research interest is in how organisms adapt to harsh environments. But what does harsh mean?
The plant I study lives in this granite outcrop that's, very, very dry. There's no water at a very early stage in the season, and there's no soil to hold on to water while the plants are growing. It's really just dependent on snow melting over the rocks and actually kind of inundating the plants for a while. And then once that's gone, the plants have about a week to either reproduce or die. So it's, it's extreme.
Mm-hmm. And so you're just kind of describing what a harsh environment is, but can you maybe talk a little bit more about what's a harsh environment for a plant?
Yes. Yeah. So for a plant, a harsh environment, I think, and how I think about it, is anywhere where you see very few plants, but you see some. That's probably a harsh environment, which is what is true of these granite outcrops. Very few species actually have colonized and live there, except for mine and some mosses. So that's one clue, is if it's just kind of depauperate of plants. And really anything that's very dry, where there's not a lot of soil moisture holding capability, is going to be difficult for a plant to thrive. So that's how I think, or very hot or very cold. So that's kind of how I think about it. But it's usually relative, this environment is very relative to the meadow right next to it, where this other species grows in a very wet, soil-rich environment. So.
Mm-hmm. What's harsh to one plant is not going to be harsh to another.
Yes.
At a different location.
Yeah. Exactly. Yeah. And, obviously once that plant has adapted to that harsh environment, can we really call it harsh anymore? Because now that plant has conquered it. Yeah. Yeah. And I mean, there's also ideas about, if you can adapt to these harsh environments, you escape other, pressures that are damaging as a plant. So you escape competition. Mm-hmm. And that's definitely true of these little guys. They no longer have to compete with other plants. They only live on mosses, really. And so they're not, there isn't interspecific competition to keep them, in their place. So they have this harsh environment, but it's they're thriving and they're dominating that environment.
I love that. Thinking about plants competing, a lot of people don't think of plants as, competing with each other, but they definitely do. For light, for water.
Yeah. I would say in some ways it's even stronger for plants because they can't move, right? Once they've dropped and germinated, they can't migrate away from their competitors. They have to, it's a dog-eat-dog, plant-eat-plant world where they're just fighting for water and light and nutrients. Those are limiting resources. So,
yeah. So then we asked, why is it important to understand why and how species adapt to harsh environments?
So that's a great question. So I think it's important for a lot of reasons. One is that we live in a changing world. So the climate is changing at a very fast rate, and understanding how plants and animals have adapted to these harsh environments sort of contemporarily can help us understand how we might be able, those plants and animals and maybe us might be able to adapt in the future with future climate change. And then there's also just, there's also more applied reasons, especially with the plants, if we find that a certain leaf shape or time that a plant flowers in the wild is adaptive to a harsh environment, we can potentially take that and put it into agricultural practices. So if we understand the genetic basis of these traits that are advantageous in harsh places in the wild, we could apply that to agriculture and have crops that can grow in more places throughout the world and populations that perhaps need the crops more. So those are sort of two, and then they're just really cool, and I think we should understand everything about our world.
Love it. Me too. So you started mentioning traits of interest, right?
Yes.
So you mentioned leaf shape. What are the traits that you're interested in and how they relate to adapting to a harsh environment?
Yeah. So with the specific plant I study, there are a few traits that if you look at them, you have reasons why you might think they're advantageous. And one of them is this monkey flower has this very distinctive lobed leaf shape. And there are physical reasons why that could actually be adaptive in a harsh, dry environment. So just the shape of the leaf can affect how temperature and water move through that plant. And that's a huge deal for whether a plant lives or dies. So that was one thing I became really interested in. And then also life history traits. So when you reproduce, when you flower, those are really important for all of us, but for plants especially in these sort of ephemeral environments where they're going to lose their water and their source of life early in the season. So they need to be able to reproduce very quickly. So early flowering time is important in these harsh habitats for plants very often. Yeah, and then I'm also interested in like how plants reproduce or how they mate. And so, some plants mate primarily with other individuals, so we can call that outcrossing. But some plants, this is a great advantage for plants is they can actually mate with themselves, and they do that predominantly. And that can be advantageous in a harsh environment because you don't have to have any other buddies, other members of your species nearby to mate with. And you don't have to rely on animal pollinators who they are not in this new harsh habitat that you used to have in your old habitat. So if you can mate with yourself, you're covered in all situations, and that's clearly helpful.
And like you said, there aren't going to be like huge densities probably of plants around in these harsh environments. So mating with yourself ensures that you're...
Exactly. Yeah, and especially if you're those pioneers, right? We think that mating with yourself is a real advantage if you're going to be colonizing new habitats because you're out there on your own. You don't have to rely on anybody else to have babies. So.
Well, especially like you said, if they are colonizing a new habitat and... flowering earlier for some reason.
Right.
Maybe there's what we call that phenotypic mismatch where like the pollinators aren't available.
So
maybe
absolutely.
they have to flower earlier to actually breed.
Yeah, absolutely. Yeah, there could be no pollinators, or now you could be flowering too early to mate with, the members of your population that live in the other environment because they flower later. So yeah, there could be a temporal disconnect there too.
So when you identify these traits that might be adaptive, what next? So how do you actually figure out if it's adaptive, if it's an evolutionarily selected for trait? Like, what are your next steps after identifying those traits?
Yeah, so I think that is the challenging part because you have so many traits diverging when a new, species colonizes a new habitat, a new population. There's all these things that are diverging, and some of those traits, like maybe flowering time, are diverging because they're selected for, but some of them are just diverging through random chance over time.
So Dr. Ferris keeps using the term diverging. What does she mean by that for our listeners who may not be familiar?
So diverging just means differentiating, changing, or moving away from each other.
And so how do you tease apart the randomly diverging from the naturally selected traits? And so there are a couple of approaches that I use. So the one I've used with my plants is to actually make genetically recombinant individuals. So to cross like a plant with a round leaf and a plant with a lobed leaf together. And then you can eventually get this hybrid population where you've mixed up the genetic backgrounds of these two parents that you cross, the round leaf parent and the lobed leaf parent. So you've got all this randomly, mixing DNA in your hybrid population. And so you can then put those randomized individuals out into a natural environment, and you can look at leaf shape, and that leaf shape is mostly isolated from other traits in the genetic background. Because of the cross you did, that leaf shape now kind of exists independent of the evolutionary history of those populations. And so you can actually ask, is it this leaf shape that's important by measuring natural selection in the field on those manipulated individuals? Or you can use fancy statistical models with genome-wide data, and you can look across the genome and you can look for genes that are showing sort of uncharacteristic patterns of association with environmental variables. So with soil moisture, with being on granite or something like that. And you could say these genes are, they're outliers, they're unusual, they're showing these weird patterns in accordance with the environment compared to the rest of the genome. So, that's a really good sign that they're doing something advantageous with those environmental variables. So I've used, both of those approaches and plan to keep using them. But it's tricky because even when you do make those genetically mixed up hybrids, you've still got, probably some, you're not having just one gene that's responsible for leaf shape independent. Like multiple traits are probably still linked and correlated in there. So it's, it's a long-term problem to solve.
So you get to study this widely distributed plant like you described, and it just so happens to be primarily distributed along beautiful elevation and moisture gradients in the Sierra Nevada mountains. Your pictures were very beautiful. Could you walk us through one of your field days, and could you tell us like what you love about being in the field and maybe what makes a hard day?
Absolutely. I can talk about all those things. So I love field work. It's wonderful that you get to be outside as your job all day. And you get to be often in beautiful places. Like I lived in Yosemite for several summers, and that was an amazing experience. And I was there for 2 to 3 months each time. But on the other hand, so a typical field day when you're planting up a large experiment like I did. So I planted about 4,000 plants and planted them like into rocks and into meadows where I had to clear plants, and so it is a lot of work. So it sounds like, oh, you're going to Yosemite for the summer. What a great vacation for you. But that's yeah, exactly, and just dance through the fields and mountains. But no. So when I was planting the experiment, and I actually planted this experiment in the month leading up to my wedding, I had not planned on getting married in the middle of my field season. But my first field experiment the year before had actually completely failed. So that is the other bad thing about field work, is that you're planning things into a natural environment. It is unpredictable. So I actually had to completely redo this very large experiment. And there were many tears that fell after the first.
So does that mean you actually planted 8,000 plants total?
Yeah. Yes. Yeah. Over 2 years. Yeah. But the first 4,000 died within a week. So I couldn't even get any data on them.
Wow.
So I feel like that's important for people to know. You can have a major failure and still get a PhD and be a scientist and continue your life as a happy person. You just have to be pretty sad for a little while. But anyways, so that can be very hard, is that it can really go south. But yeah, when it did work, so a typical day leading up to my wedding is that my husband was my field assistant, and we would go out for 13 to 15 hours. And that was planting. I had to drive about 2 hours to get to my sites, and then spent about 13 to 15 hours planting tiny seedlings that are like half the size of your fingernail into rocks with forceps on your knees. And it's very hard work. And then some of them wash away and you have to replace them. and then you get married in St. Louis, and then you fly back out to California for 2 months and don't see your husband until the end of your field season again. But overall, I mean, it's incredible. It's this beautiful environment, and you get this sort of insider's experience of it because I was living there. I was living in this house full of 13 other field biologists. So, I was like 28. I had just gotten married, and then I went back to college for 2 months, basically, with these awesome biologists, and they were like my field family, and they helped me census my plants. But they're long days, and it's physically demanding. It's kind of like farming on a very weird plant scale that nobody would ever be able to eat. But it is a lot of labor, and you're not sure how it's going to turn out. So the field season that did actually work for me, I still had some unexpected twists and turns where a huge fire happened in Yosemite at the end of my field season, and I actually got cut off from one of my field sites, and I luckily collected most of my data there, but it burned, and I didn't get to collect all of my data. And I also just couldn't go into 2 of my field sites for a month.
Was the rim fire?
Yes, it was the rim fire, the Great Rim Fire of 2013.
Part 3, mice. How did you transition from plants to mammals?
Yeah, that's a great question. It is an unusual transition.
Mm-hmm.
So it's really because of my PhD advisor. I will blame it on him. So he was a great advisor, and the advice he gave to his students towards the end of their PhD about their postdocs was that you should either ask a very different kind of question in a similar system, a similar organism, or you should ask a similar question in a very different organism so that you can show that you have this breadth and that you have intellectual curiosity and you're willing to learn new things and, just basically to show like who you can be as a scientist over your career, which I think is good advice. I went like a little bit farther away than most people. Yeah. So like we'd had people from the lab like go on and work with fruit flies, in their postdocs. Actually, a couple people did that, but asked similar questions about the genetics of adaptation. And while flies are certainly different than plants, they're a little bit more manageable than mice. So I think I took that advice to the most extreme.
So in mice, what adaptive traits were you interested in? And then what can those traits tell us about adaptation to harsh environments?
Yeah, that's a great question. So we had some preliminary evidence that mice in North America in, just a couple hundred years had already sort of differentiated in their body size, where mice that were in the north in cold environments were larger than mice in the south in hot environments. And that follows what we call Bergmann's rule, where we think that endothermic animals should be larger in colder environments, basically because they can hold on to their body heat better if they're larger. They have smaller surface area to volume ratios. So they're keeping that body heat, and that should keep them through the winter. So we thought that should be different among these mice. And when we looked at the phenotypic data, it was. So mice in the north are significantly larger than mice in the south. So they're holding on to that heat. And then there was a paper published in the 90s that had collected wild mice from the east coast of North America, and mice from New York were building bigger nests even after a generation in the lab than mice from the south. And this is really cool because it's a behavior, but it seemed to be genetically determined because it was still happening even after you brought wild mice and let them breed for one generation in the lab. Mm-hmm. And it's also a clearly adaptive phenotype. If you can build a larger, better insulated home for yourself, then you're clearly going to stay warmer through these cold, New York winters. And so we also measured nest building in all of these mice collected from 5 populations kind of across North and South America, and we found that mice in northern cold populations do build significantly larger nests even after up to 5 generations of being bred in the lab. So it's a clear genetic difference, and it's genetically determined behavior, which I just think is super cool. And there's actually another behavior that we ended up measuring, which is also genetically differentiated, and that's activity, wheel running activity. So we attached bike odometers to little mouse wheels and tracked how far a mouse could run and how fast in a 24-hour period. And the ones in the north, in the cold environments, run about twice as far in the same period as the ones from the south. And this is linked to metabolism. So we think that more active mice have higher metabolisms, and this is true in humans too, and cold-adapted mammals have higher metabolism, and that's linked to activity. So that's something that, if you're churning out more energy, you're going to be staying warmer, and you're going to need to do that if you're in a colder environment, because the environment is much further away from your body temperature. So you're going to need to spend a lot of energy to keep your body temperature at, 98.6 when it's 30, than if it's, 95. So
Fahrenheit.
Yeah, just to be clear, those were in Fahrenheit.
Could you briefly, because we loved it so much, describe how you tested the differences in nest building?
Yeah, absolutely. So we did this very sophisticated assay where we put about 40 grams of unused medical grade cotton on top of a mouse's cage. And so they have these wire tops on the cages. So the mice actually will reach up with their little mouse hands and pull the cotton down into the cage. And so it's really cute to watch. Actually, they're standing on their hind legs and they're pulling cotton through a wire mesh. And the ones from New York will fill their cage so full that you can only see one little mouse window with a nose peeking through. And most of the time the mouse is clearly hiding in all of its tunnels that it's made through that cotton. Whereas the ones from southern populations just kind of toss the cotton around willy-nilly. It's not, they haven't created this very structured and insulating nest. So it's a pretty adorable phenotype and clearly helpful, to stay warm.
Cool to see that actual cool, behavioral difference.
Yes,
that has
yeah.
genetic component.
It
So
is.
you see these behavioral differences in these mice. You see that their body size is very according to these latitudinal gradients from north to south. You're seeing it in their behavior. And so then what's the next step? What do you do next?
Yeah, so the next step is, we want to know at the genomic level, are they adapting? What genes are under selection? And are some of those genes that are under natural selection involved in these phenotypes that we see being different? And so we use these genomic tools and statistical models to look for associations between areas of the genome and temperature across these, large latitudinal transects. And we looked for just really tight associations between certain genes and this environmental variable of temperature that looked very different from the rest of the genome. And those sort of outlier or, special loci, those are the ones that are under natural selection. And we find that some of those loci that are under the strongest selection, that are showing the tightest associations with mean annual temperature, some of them are actually involved in body size and body weight. And so things like that are really cool. And some of them are involved in exercise, related metabolism too. So it's exciting to see that.
And like you said, mice are so well studied for medical purposes. So we already know a lot about their genomes and what these different genes are doing. So.
Yeah, that's one of the powerful things is there's been a lot of experimental work done, at the genetic level in mice where they've, actually knocked out and made unfunctional a lot of these genes, and then you see what happens to the mouse. So we know that if you knock out this one gene, MC3R, melanocortin receptor 3, mice get really fat. And then we saw a pattern in our data that suggested that gene is under very strong selection. And then we see this strong climb in body size. So that's all coming together very nicely. I feel like we've found, things that are under selection, and we also know what they do. They affect ecologically important traits. So that's exciting.
Part 4 of Mice and Mimulus. So you study the genetics of adaptations to harsh environments using 2 species that are very different, a common mouse and a common annual flower. How are these 2 systems similar, and how are they different?
That's a great question. So they're similar in that they both have a lot of genetic tools, which is important for what I do. So this annual flower, Laciniatus, is very closely related to this seep yellow monkey flower, Mimulus guttatus, which has a fully sequenced genome. So that means that I basically have a fully sequenced genome to work with because they're so closely related, which is awesome. And then, of course, the house mouse is the biomedical model system, and there's an amazing, genomic resources there. And you can also ask questions about adaptation to very different environments in both of them. So because house mice occur all over the world in all sorts of different climates, they've had to adapt to lots of different things. And then monkey flowers have, on a smaller scale, also really sort of radiated into all these very diverse habitats, old mines, granite outcrops, very harsh plant habitats, even though they occur very close to each other geographically. So you can ask the same questions about how do organisms, when they're encountering new and stressful environments, how do they change and adapt, and what is the genetic basis of that? But I did just mention one difference is that the mouse work is really on this very large climatic scale, so sort of across continents and looking at climatic change on that level, whereas the monkey flowers are occurring within meters of each other, but in dramatically different soils, and so they're still experiencing dramatically different environments. So I like being able to span both of those geographic scales. It's I love that things right next to each other can be really different in terms of how they're evolving and what they're having to adapt to, but it's also nice to see this broader range of change across a climatic gradient on a continent. They're also different in that one of them moves and one of them does not, and that makes a huge difference. So, I came from a plant background and then did my first postdoc at UC Berkeley in a mouse lab, and I was coming into it as I'm driven by these questions of how do organisms adapt to harsh environments, and so I should be able to ask those just as easily in mice as in Mimulus. But it is a little different because mice run away from you and they bite you, and yeah, they're mammals, and mammals are not plants. So that I hadn't thought about the organismal differences as much, which may sound weird to everyone who hears that I've worked on both monkey flowers and mice, but in theory, you can do evolutionary biology with anything and ask the same questions. But in practice, it is different. It is more challenging to work with things that move and where there's a lot of regulations over what you can do with them, because everyone gets very upset if a mouse gets injured. If a Mimulus gets injured, no one cares. Like, nobody is calling me at night being like, your monkey flower lost its leaf. I know, it's very insensitive, really. Exactly. You still can't pick any mouse, no matter if you're a botanist or not. Yeah, it doesn't apply.
So kind of continuing off of that, you said that your main question was, studying adaptation to harsh environments. How did you even come up with that question? What kind of drove you into that specific question?
So I think that's just something. So I've always really liked, because I've admired organisms that occur in places where they should not. Like, I love desert plants because it's like, what are you doing here? Like this, like nothing should be able to grow in this environment, and yet you have. And so maybe that's the story aspect of it that I just find really compelling, that like life will find a way.
So you studied adaptations to harsh environments in plants and in mice. Could you study this in humans?
People do, absolutely. You can totally study this in humans. So it's much easier to do the genomic work. Humans are hard to plant into the ground, and you don't want to make them mate and recombine their genomes. That's unethical. But you can do a lot of really cool things with, human genome data. And there's a lot of it out there because of the biomedical field really wanting to generate more genome data across populations and across the world. So people are absolutely looking for adaptation to different diets in human populations. That we've found genes underlying, lactase persistence, so how humans can keep drinking milk in some populations, and that's actually evolved multiple times, and there are different genetic changes involved in it, although it's the same genes sometimes. So yeah, like this is totally applicable to humans, although we won't make them mate. They're a terrible system for that anyways, because we only make like 2 progeny? That's awful. You want at least 100 out of a single mating. So
Add more babies. So just at a personal level, from a scientific standpoint as well, though, what excites you most about your work?
Yeah, well, I think maybe I said this earlier, but I really like understanding how things work. And so that's one of the things that drew me to biology. But I also, in addition to majoring in biology as an undergraduate, I was an English literature major. So I got 2 BAs or whatever. And I really love stories. And so I think, understanding how things work and being able to tell this story of how things have become the way they are, because that's really what evolution is, right? Like, how did we get to this point? How did the world and all the organisms in it get here? That's just really exciting to me, is being able to, I piece together these small stories about a certain plant and a certain environment, but overall, it's really like the origins story of all of us. So I just think that's really exciting. Also, mouse nests, and leaf shape, because it's beautiful.
Flowers.
Plants.
Mimulus.
Mice.
This has been RadioBio with Dr. Katie Ferris. Thanks for listening.
RadioBio is supported by the Quantitative and Systems Biology Graduate Group and the Graduate Division at the University of California, Merced. For more information, you can visit our website at radiobio.net or follow us on Facebook, Twitter, or Instagram.